Okay, so welcome to Biochemistry and Cell Biology, and in this presentation I'll be covering glycolysis. So the learning objectives are to be able to define redox reaction, to know the terms oxidation and reduction, to know where in the cell glycolysis takes place, to know that ATP is the main energy currency of the cell, to be able to name the two common electron carriers and identify their oxidized and reduced forms, to be able to outline glycolysis and know the essential details of the three stages, to know the structures of glucose and pyruvate, and to be able to identify phosphorylation, isomerization, and oxidation reactions. So here's a few definitions to start us off with. So oxidation is the loss of electrons, and reduction is the gain of electrons. So as you're all probably aware, there's a little mnemonic to help you remember this, and that's oil rig. So oil rig stands for oxidation is loss, reduction is gain. And as you know, we're talking in terms of electrons. So then redox, which is a coupled reaction in which one species is oxidized and another is reduced. And then glycolysis, which is the metabolic pathway which converts one molecule of glucose into two pyruvate molecules. So this here is just a basic overview of glucose metabolism. So essentially there's three main steps. You've got glycolysis, the citric acid cycle, or TCA cycle, or the Krebs cycle, and oxidative phosphorylation, or the electron transport chain. And in this presentation we're just going to be covering this first step here, which is glycolysis, which is going from glucose to pyruvate. So just before we even start to talk about glycolysis, there's two electron carriers which are important throughout all of glucose metabolism. And those two electron carriers are NAD and FAD. So this here is NAD, and then when it's going to its reduced form it's called NADH, or you can call it reduced NAD. And the only difference is that this molecule then has hydrogens attached to it, only one hydrogen here on this carbon here. Then over here we've got FAD. So this here is the normal form of FAD, and this here is the reduced form, and as you can see we've got one hydrogen, two hydrogens attached. So the reduced form of FAD is FADH2. Okay, so now we can start talking about glycolysis. So step one, glycolysis of glucose metabolism. So glycolysis itself contains of three main steps. The first of which is in the investment stage. So here we are investing two ATP molecules to phosphorylate the glucose, and this will help facilitate the cleavage of glucose. We then have the cleavage step. This is the splitting of one molecule of fructose 1,6-bisphosphate into glyceraldehyde 3-phosphate, also known as G3P. And also this also forms dihydroxyacetone phosphate, which then has to get isomerized to form the second molecule of G3P. Then the final step is the energy harvest. So in the energy harvest, this is the conversion of G3P into pyruvate, which also results in two ATPs and one reduced NAD per molecule of G3P. So let's talk about the investment stage. So here we are going from glucose into fructose 1,6-bisphosphate. So just so you're aware, anything that's highlighted in red is the enzyme which facilitates the reaction. So the first step of an investment, we are phosphorylating glucose. So glucose plus ATP goes into glucose 6-phosphate and ADP, and this is catalyzed by hexokinase. Then that glucose 6-phosphate is then isomerized into fructose 6-phosphate, and that's done by phosphoglucose isomerase. And then that fructose 6-phosphate is then further phosphorylated with ATP to form fructose 1,6-bisphosphate and ADP, and that is catalyzed by phosphofructokinase, or you can abbreviate that to PFK. So now that we've phosphorylated the glucose, we're now ready to cleave it. So literally imagine an axe cutting through a chunk of wood, splitting it into two. So the fructose 1,6-bisphosphate is cleaved into glyceraldehyde 3-phosphate, or G3P, and dihydroxyacetone phosphate, and that's catalyzed by the enzyme adolase. However, we need G3P in order to follow through into the energy harvest, so the dihydroxyacetone must first be isomerized by the enzyme triphosphate isomerase to form the dihydroxyacetone phosphate into the G3P. And now we're on the final step. This is the energy harvest. So note, if we go to the previous slide, you you can see that the fructose 1,6-bisphosphate is split into two G3Ps, okay? So anything that happens during the energy harvest happens twice per molecule of glucose, because if you remember, we get two G3Ps per molecule of glucose. So the glyceraldehyde 3-phosphate is going to be converted into pyruvate. However, as we know in biology, it's just not one straightforward reaction. There's several steps involved to get us there. So first of all, the G3P and inorganic phosphate and NAD phosphorylates the G3P to form 1,3-biphosphoglycerate and reduced NAD. And that's catalyzed by the enzyme glyceraldehyde 3-phosphate dehydrogenase. The 1,3-bisphosphoglycerate and ADP is then converted into 3-phosphoglycerate and ATP. Note we are now getting some ATP, so we're harvesting some energy from this. That's catalyzed by phosphoglycerate kinase. The 3-phosphoglycerate is then isomerized to 2-phosphoglycerate by the enzyme phosphoglycerate mutase. The 2-phosphoglycerate is then turned into phosphoenolpyruvate and water, and that is catalyzed by enolase. And then finally, the phosphoenolpyruvate and ADP turns into pyruvate and ATP, which is catalyzed by pyruvate kinase. So now that's a brief overview of the three main stages. So now if you just look at what we've actually done. So here on the left we can see the two forms of glucose. We've got the linear form, note that this is showing where this oxygen is, and the cyclic form. So we've just converted just one molecule of glucose into two molecules of pyruvate. And just so you're aware, try and make sure you're able to draw all these, because it will help in understanding what's going on in the reaction. Okay, so now if we compare before and after glycolysis, so before we have two ATP molecules, two NAD molecules, and one molecule of glucose. Then once we put them through glycolysis, we end up with 4 ATP molecules, 2 NADH molecules, and 2 pyruvate molecules. And please note, the net yield of ATP is only 2, not 4. The reason for this is, yes, we get 4 produced, but if you remember correctly, 2 were required to invest to phosphorylate the glucose. Alright, so just remember the net yield is 2 ATP molecules. Now we come to the end, so now we're at the test yourself section. So here I'm going to ask you 3 questions. I've allocated a number of marks for that, and I want you to try and figure out the answers. I'm not going to tell you the answers, the marks are there just to guide you to how many points you need. So the first question for 3 marks, what are the 3 main steps in glycolysis? For 1 mark, which enzyme is used to convert 1,3-biphosphoglycerate into 3-phosphoglycerate? Okay, so you do need to make sure you remember those enzymes. And finally, an essay style question for 25 marks, outline the entire process of glycolysis, including the reactants, products, and enzymes used to catalyze the reactions. So there we are, I hope you enjoyed this. I know it's been a while since I've released a video, but I've recently got some lip piercings and I was no longer able to talk. So now that I'm able to talk, I'm able to fully push these all out and ready to time for the exams. So thank you for watching, I hope you enjoyed, and good luck revising, guys.